How can a single change in a DNA sequence shape human survival across regions with vastly different environmental pressures? Sickle cell anemia offers a clear example of how one altered nucleotide within the HBB gene influences both protection and vulnerability. The mutation modifies hemoglobin structure, creating red blood cells that deform under low oxygen conditions. This deformation leads to circulatory challenges, yet also interferes with the life cycle of malaria-causing parasites. Examining the condition across populations highlights how its distribution aligns with climates where malaria was historically prevalent, reflecting long-term interactions between human biology and environmental pressures. Through molecular mechanisms, ancestral migration patterns, and regional prevalence, sickle cell anemia illustrates how a single genetic variation can shape health outcomes across generations. The foundation of sickle cell anemia lies in a single nucleotide substitution within the HBB gene, which encodes the beta chain of hemoglobin. This substitution replaces adenine with thymine, altering one amino acid in the hemoglobin protein. Although the change appears minor, it modifies the protein's behavior under specific physiological conditions. Hemoglobin molecules containing the altered sequence tend to polymerize when oxygen levels fall, forming rigid structures that distort the shape of red blood cells. The mechanism centers on how these polymerized hemoglobin strands influence cell flexibility. Normal red blood cells move smoothly through capillaries, maintaining efficient oxygen transport across tissues. However, cells containing the altered hemoglobin become rigid when deoxygenated, creating the characteristic sickle shape. These deformed cells can obstruct narrow vessels, reducing blood flow and causing localized oxygen shortages. Evidence from cellular studies shows that repeated cycles of sickling and unsickling weaken cell membranes, shortening the lifespan of affected cells. Now, the distribution of this mutation varies across populations. The allele is most common in regions where malaria historically posed a significant selective pressure. Individuals carrying one copy of the mutation experience improved resistance to severe malaria, creating a heterozygote advantage that supports the allele's persistence. By comparison, individuals with two altered copies experience the full clinical expression of sickle cell anemia. Today, population genetics provides insight into how this variation spread through ancestral groups. Patterns of migration, gene flow, and demographic change shaped the allele's presence in Africa, the Mediterranean, the Middle East, and parts of India. These historical processes reveal how a single nucleotide change can influence both biological function and population-level adaptation. This chapter outlines the molecular foundation of the condition and establishes the link between genetic structure and environmental context. The physiological consequences of sickle cell anemia arise from the structural behavior of hemoglobin under low oxygen conditions. When hemoglobin S polymerizes, red blood cells lose their normal flexibility, forming rigid shapes that travel poorly through small vessels. This change disrupts circulation within capillary networks, creating localized reductions in oxygen delivery. The resulting stress affects tissues that rely on continuous blood flow, particularly within the spleen, bones, and vascular regions that experience frequent pressure fluctuations. The central mechanism involves repeated cycles of deformation. Sickled cells regain shape when reoxygenated, but the process weakens their membranes over time. Evidence from hematological studies shows that these cells have significantly shorter lifespans, contributing to chronic anemia as the body struggles to replace them. In contrast, normal red blood cells maintain structural stability across multiple circulation cycles, supporting consistent oxygen transport. Symptoms vary across individuals and populations due to differences in genetic background, environmental conditions, and the prevalence of protective modifiers. Factors such as fetal hemoglobin levels influence disease severity by reducing the extent of polymerization. Today, molecular research highlights how interactions between genetic variants affect the expression and progression of the condition. Geographic patterns offer additional context. Regions historically affected by malaria show higher carrier frequencies, reflecting the selective advantage of heterozygosity. However, clinical outcomes differ in areas with limited medical access, creating variation in how the condition manifests across populations. These differences illustrate how biological mechanisms and environmental settings shape the lived experience of the disorder. In summary, the physiological consequences of sickle cell anemia reflect the combined effects of hemoglobin, polymerization, restricted blood flow, and variability in genetic modifiers. This framework provides the biological foundation for understanding how the condition influences health across diverse environments. The global distribution of sickle cell anemia reflects a long-standing relationship between human populations and environments shaped by malaria. The allele responsible for the condition is most prevalent in regions where malaria transmission was historically intense, including parts of sub-Saharan Africa, the Mediterranean basin, the Middle East, and South Asia. In these areas, the heterozygous state offered a survival advantage by reducing the severity of malaria infection, enabling the allele to persist and spread within local populations. Evidence from population genetics highlights clear geographic clustering. The highest frequencies occur in West and Central Africa, where malaria transmission rates historically reached their peak. By comparison, lower but notable frequencies extend into regions such as Greece, southern Italy, the Arabian Peninsula, and western India. These patterns align with ecological zones where climate-driven conditions supported the life cycle of malaria-causing mosquitoes. Now, the distribution also reflects historical migration and gene flow, movements during the trans-Saharan trade, interactions between Mediterranean populations, and expansions along the Indian Ocean. Trade routes contributed to the spread of the allele. More recent global migrations, including movements during the Atlantic slave trade and contemporary population shifts, have introduced the allele into regions such as the Americas, Europe, and parts of the Middle East, shaping modern prevalence profiles. Environmental pressures remain a central factor. As malaria persisted in tropical and subtropical climates, selective pressure maintained the allele within populations exposed to the disease. In contrast, regions without malaria show lower frequencies, illustrating how local ecological contexts influence genetic distribution. These patterns demonstrate how ancestry, environment, and historical migration interact to shape the global presence of the sickle cell allele. This combination of selective pressure and demographic movement provides a clear framework for understanding its geographic and population-level variation. The evolutionary context of sickle cell anemia centers on the balance between protection and vulnerability, created by a single nucleotide substitution. Individuals carrying one copy of the allele experience reduced severity of malaria infections, a clear advantage in regions where the disease historically caused high mortality. This heterozygote advantage allowed the allele to persist, creating a selective equilibrium in which both protective and harmful outcomes coexist within populations. The dynamic illustrates how genetic variation can remain stable when environmental pressures favor intermediate forms. Mechanistically, the advantage arises because sickled red blood cells provide a less favorable environment for the malaria parasite. Evidence from parasitological studies shows that infected cells containing hemoglobin S tend to be cleared more efficiently by the spleen, reducing the parasite's ability to replicate. In contrast, individuals with two copies of the allele lack this balance and experience the full clinical expression of sickle cell anemia. Now, population-level frequencies reflect how selective pressures interact with demographic history. Regions with sustained malaria transmission show higher allele frequencies, while areas without these pressures exhibit much lower levels. These differences correspond to patterns of ancestry, where lineages from malaria-endemic regions maintain higher carrier rates. By comparison, populations with ancestral origins in cooler or high-altitude environments display minimal frequencies, reflecting limited exposure to the parasite. Intergenerational dynamics shape how the allele persists. As populations migrate or intermix, gene flow introduces the mutation into new regions, altering local prevalence. Today, global movement continues to reshape distribution patterns, creating populations where the evolutionary trade-off appears in new contexts without the original environmental pressures. In this framework, sickle cell anemia represents a clear example of how selective pressures, genetic variation, and long-term population history interact to create enduring patterns within human lineages. Modern research on sickle cell anemia focuses on understanding how genetic variation influences clinical outcomes and how early detection improves long-term health. Advances in molecular biology have clarified how hemoglobin S polymerization interacts with cellular pathways, revealing factors that shape disease severity. Variants that increase fetal hemoglobin production reduce polymer formation, offering partial protection and creating measurable differences among individuals with the same underlying mutation. These mechanisms help explain why symptoms vary across families and populations. Today, genetic screening plays a central role in clinical management. Newborn screening programs identify affected individuals before symptoms appear, enabling early interventions that reduce complications. Evidence from population-level studies shows that early detection lowers mortality and improves quality of life through timely vaccination, infection prevention, and monitoring of organ function. By contrast, regions without widespread screening often experience later diagnoses, contributing to more severe outcomes. Now, global health initiatives aim to expand access to testing, particularly in countries with high carrier frequencies. These efforts include community-level programs, improved laboratory infrastructure, and educational campaigns that highlight the genetic basis of the condition. Migration patterns create additional considerations. As individuals from high prevalence regions move into areas where screening has not traditionally been prioritized. Public health systems increasingly adapt to these demographic changes by integrating targeted testing and counseling. Genomic technologies continue to refine diagnostic accuracy. Sequencing methods identify rare variants, clarify ancestry-related differences, and support research into genetic modifiers that influence disease expression. These insights contribute to personalized approaches that consider both genetic background and environmental factors. Collectively, modern research and screening practices demonstrate how scientific understanding and public health strategies intersect. This chapter provides a foundation for examining emerging therapies and long-term management approaches in the context of global population change.